everyone, a very good morning to all of you. This is Dr. Shal Pragya and today we will talk about seed structure and germination. In this video we will study about seed, and as we know seed we have studied in previous classes also, but here we will study in little bit detail. So more information we will get about seed, and then we will study about classification and the structure of different types of seeds. So let's start the topic with the seed structure and germination. But before we talk about the structure and germination of seed, we should discuss about some terminology such as fruit, seed and grain. About fruit and seed we know and we have studied in previous classes also, but grain is little bit new word for us, and many of us are confused with seed and grain. Some students, they think that seed and grains are the same thing, but it's a wrong concept. Seed and grains are different from each other. How they are different we will study in this video. So let's start with fruit. So fruit is the enlarged ripened ovary. About fruit, one complete video I made. So if you have any confusion, you watch the previous video. Fruit protects the seed and helps in seed dispersal. Seed is the ripened ovule. What is seed? It is the ripened ovule. It contains an embryo which develops into a new plant. So seed coat protects the embryo from mechanical damage. Embryo develops into new plants. Example is bean seed, peas or many other seeds are there. Now we will talk about grains. So grain is the combination of fruit and seed. It is a combination of fruit and seed because in grains the fruit wall that develops from the ovarian wall, fruit wall जो ovarian wall से बना होता है, and the seed coat that is formed by the integuments of the ovule. Ovule के integuments बाद में क्या चीज में convert कर जाता है? Seed coat में. तो अब ग्रेन की अगर बात आती है तो ग्रेन इज द कॉम्बिनेशन ऑफ फ्रूट एंड सीड because in grains the fruit wall and the seed coat are fused together to form a protective layer. Example is maize, wheat. Now we will get some more information about seed. So as I told you, seed is a mature ovule that is formed after fertilization. It contains a tiny living plant in it that is known as embryo. Embryo consists of a radicle. Radicle is the future root. It is the future root. R for radicle, R for root system. So radicle will give rise to root system in future, and plumule is the future shoot. And it also consists leaf-like structure that is known as cotyledons or seed leaf. The embryo remains in inactive or dormant stage until exposed to favorable condition. The seed also contains food material for the nourishment of the embryo during germination that is known as endosperm. इंडोस्पर्म के कंटेंट इतने ज्यादा होते हैं कि कुछ सीड 100 इयर्स और कह सकते हैं कि कुछ सीड तो 1000 इयर्स तक भी डॉर्मेंट स्टेज में रह सकते हैं. सीड्स वेरी इन साइज. अगर साइज की बात करें तो सीड वेरी इन साइज. कुछ तो इतने स्मॉल होते हैं कि बड़े मुश्किल से नेकेड आई से देख पाते हैं. फॉर एग्जांपल पॉपी सीड्स, स्ट्रॉबेरी सीड्स, और कुछ सीड्स जो मीडियम साइज के होते हैं जैसे मैंगो सीड जो हम लोग देख पा रहे हैं तो वो मीडियम साइज के हैं, और कुछ सीड जो बहुत बड़े होते हैं. तो लार्जेस्ट सीड की अगर बात करें तो लार्जेस्ट सीड है कोकोनट और डबल कोकोनट. The shape, size and structure of seeds differ. इनके शेप, साइज और स्ट्रक्चर कैसे होंगे ये कंसिडरेबली डिफर करते हैं डिफरेंट प्लांट्स में, बट द बेसिक स्ट्रक्चर ऑफ मोस्ट ऑफ द सीड इज सेम. सो नेक्स्ट वी विल टॉक about the different types of seeds and then different structure of different types of seeds. Now we will talk about types of seeds. On the basis of number of cotyledons, seeds are classified into monocotyledonous seeds and dicotyledonous seeds. So mono, as the prefix mono is suggesting, mono means one. So monocotyledonous seeds contain only one cotyledon, whereas dicotyledonous seeds contain two cotyledon. Example of monocotyledonous seed is maize, grass, whereas example of dicotyledonous seed is pea, gram, beans, and many other examples are there. Now we will talk about the classification of seeds on the basis of endosperm. On the basis of endosperm. So if the seeds contain endosperm, then it is known as endospermic seed. Endospermic seeds are also known as albuminous seeds. Whereas if the endosperm is absent, then it is known as axalbuminous seed. It is known as axalbuminous seed or non-endospermic seed. In endospermic seed, as we know, endosperm is present, and if the endosperm is present, then the cotyledons are thin and membranous in such seeds, because the function of cotyledon and endosperm is more or less same. So if endosperm is present, then the seed cotyledon is thin and membranous, and on the basis of number of cotyledons present in the endospermic seeds, these are further divided into monocot albuminous seeds and dicot albuminous seeds. In monocot albuminous seeds, only one cotyledon is present, that is thin and papery cotyledon. Examples are cereals, millets, palm, whereas in dicot albuminous seeds, two cotyledons, two thin and papery cotyledons are present, and examples are poppy, custard apple. Now we will talk about exalbuminous. Exalbuminous means non-endospermic seeds. So in this type of seeds, इसमें क्या होंगे? एंडोस्पर्म एब्सेंट होंगे या तो एंडोस्पर्म बहुत कम डेवलप्ड होंगे. इसमें जो कॉटिलिडन्स होंगे वो कैसे होंगे? थिक होंगे, फ्लैशी होंगे. सो we can say that in such seeds the cotyledons store food and become thick and fleshy. This type is also further divided into monocot axalbuminous seeds, and the examples of monocot axalbuminous seeds are Vallisneria, orchids, amorphophallus, and the examples of dicot axalbuminous seeds are gram, pea, mango, mustard. Now we will study about the structure of seeds. For this we will talk about one example of dicotyledonous seeds and one example of monocotyledonous seed. In dicotyledonous seed, we will take the example of bean seed, whereas in monocotyledonous seed, we will take the example of maize grain. There are different kinds of bean seeds, but the basic structure is same. Most of the bean seeds are kidney shaped with a convex and a concave side. इसके एक तरफ convex side होगा, दूसरी तरफ concave होगा. Seed is protected by testa, that is the outermost hard brownish covering. जो उसका बाहरी वाला लेयर होगा, प्रोटेक्टिंग कवरिंग होगा, दैट इज टेस्टा, एंड टेस्टा इज वेरी हार्ड एंड टफ कवरिंग. इट प्रोटेक्ट्स द सीड फ्रॉम द external injury or pathogens. So we can say that this is the tough covering, outermost covering, and it protects the delicate inner parts of the seed from injury and pathogens. For providing extra protection, one another covering is there, that is known as tegmen. तो टेस्टा के अलावा एक और कवरिंग होती है जिसे हम लोग टेगमेन कहते हैं. टेगमेन इज अ थिन लेयर लाइंग नेक्स्ट टू द टेस्टा. टेस्टा के ठीक नीचे एक थिन लेयर होती है जिसे हम लोग टेगमेन बोलते हैं. नेक्स्ट स्ट्रक्चर दैट वी हैव टू स्टडी इज हाइलम. So hilum is a whitish oval scar on the concave side, जो अंदर की ओर धसी हुई साइड होगी, वहां पर एक ओवल शेप्ड स्ट्रक्चर पाई जाएगी जिसे हम लोग क्या कहते हैं, हाइलम. So this is the spot where the ovule was attached to the ovary wall through placenta. यही वो जगह है जहां से कि ओव्यूल अटैच थी किससे, ओवरी वॉल से, और ये जो अटैचमेंट हुई थी वो किसके थ्रू हुई थी, प्लासेंटा के थ्रू. Next we will talk about micropyle. Micropyle is a tiny pore situated near the हाइलम. ये हाइलम के नजदीक एक और टाइनी पोर होती है जिसे हम लोग माइक्रोपाइल बोलते हैं. This is the opening through which the pollen tube had entered into the, पॉलन ट्यूब कहाँ एंटर करेगी, ओव्यूल में. यही वो जगह है जहां से पॉलन ट्यूब ने एंटर किया था, ओव्यूल में. सो माइक्रोपाइल परफॉर्म्स टू फंक्शंस. माइक्रोपाइल परफॉर्म्स two function. The first is absorption of water during germination or while soaked in water. जब इसे पानी के अंदर डालेंगे तो जो water absorbed होगा through this pore only. Now the next function is that this is the pore where diffusion of respiratory gases occurs for the growing embryo. Below the seed coat, there are two thick cotyledons that provide nourishment or protection to the embryo. When we separate the cotyledons, we find a tiny embryo attached to one of the cotyledon. In the structure of embryo, radicle and plumule that we have already discussed in the starting of this video, but we have not discussed epicotyl and hypocotyl part of the embryo. So as the prefix suggests, epi means above, cotyl means cotyledon, whereas hypo means below and cotyl means cotyledon. So epicotyl is the region of the embryo between the point of attachment of cotyledons and the plumule. Cotyledons और plumule के बीच का जो पोर्शन होगा, कॉटिलीडन से ऊपर की ओर जब एम्ब्रियो बढ़ेंगे तो वो प्लूम्यूल होगा. अब कॉटिलीडन और प्लूम्यूल के बीच में जो स्पेस होगा उसे हम लोग कहेंगे एपिकॉटिल, whereas the space between the attachment point to the radicle is known as hypocotyl. तो hypocotyl हमेशा नीचे की ओर ग्रो करेगा और hypocotyl will continues to radicle, whereas epicotyl will continues to plumule. Now we will discuss the structure of a monocotyledonous seed, and for that we will take an example of maize grain. अब मेज सीड को मेज ग्रेन क्यों कहा गया, ये तो मैंने स्टार्टिंग में आप लोगों को बताया था. In the maize grain, the testa and the pericarp, that is also known as fruit wall, are fused together to form a protective layer. Micropyle and hilum is not visible because of presence of pericarp, which was visible in the bean seed. Major part of the grain contains a large endosperm, which is the food storage tissue. On one side of the grain occurs a small embryo. तो एक कोने में क्या होगा, एक छोटा एम्ब्रियो पाया जाएगा, which is separated from the endosperm by a thin epithelial layer. A protein-rich alluron layer covers the endosperm, और endosperm चारों तरफ से सराउंडेड रहेगा किससे, एक प्रोटीन लेयर से, that is known as alluron layer. The embryo consists of two parts, that is radicle and the plumule. The maize grain consists of only a single cotyledon, that is known as scutellum here. यहाँ पर cotyledon को क्या कहेंगे, scutellum. The radicle is the lower pointed part and it is covered by a protective sheath that is known as coleorhiza. That is known as coleorhiza. Whereas plumule is present in the upper broader part which is enclosed by coleoptile. तो coleorhiza को किसे cover करेगा, radicle को, and coleoptile will cover the plumule part. So on the basis of this description, now you can differentiate between the maize grain and the bean seed. So I hope you would have enjoyed the video. Now it's time to summarize that what we have studied in this video. So In this video, we have studied classification and the structure of seed. Then we have studied structure of bean seed, then structure of maize seed, and afterwards we have understood the differentiation between the maize grain and bean seed. So see you very soon in my next video, that is about germination of seed. So thank you for watching the video.